Hello everyone. So the rest 12 contents of microbiology record, second year record, I'm doing as a part 2. So yeah, let's go through it. So animals pathogenicity test to test the pathogenicity of microorganisms by inoculation into laboratory animals such as rabbit, guinea pigs, etc. So yeah, this is the principle and the requirements. Yeah, specimen we take is different bacterial suspension experimental animals. So coming to the procedure, here guinea pigs, so intramuscular inoculation is done in guinea pigs. So yeah, whereas coming to the rabbits, intravenous inoculation is used. Coming to rats, intraperitoneal inoculation is done. So coming to the mice, intranasal inoculation is used. So the 14th content is biochemical reactions in identification of bacteria. So what are the biochemical reactions we will see? So sugar fermentation test to test the production of acid and gas by the fermentation of given substance. So yeah, this is the principle. Yeah, inverted Durham's tube showing gas production. Yeah, this is a picture of sugar fermentation test, so using Durham's tube method. So yeah, so positive test shows pinkish red color, whereas negative yellow to colorless. So these are the examples of fermentive bacteria. So the second biochemical reaction is indole test to detect whether the bacteria can utilize tryptophan provided to it. So this is the negative test, this is the positive, where we can see the red color ring. So this indicates indole positive. This is the reaction of indole test. Yeah, principle and procedure. So yeah, these are the positive and negative for negative bacteria for indole test. So the third one is urease production to determine the ability of an organism. So here principle is to determine the ability of an organism to produce an enzyme urease which splits urea to ammonia. So ammonia makes the medium alkaline and thus phenol red indicator change to pink or red in color. So this is the positive test and this is the negative. So yeah, this is the procedure, reagents and interpretation. So these are the urease positive and these are the urease negative bacteria. So coming to citrate utilization test to determine the ability of the bacteria to utilize citrate as a sole source of carbon for its growth. So yeah, so citrate utilization test, this is a picture I've drawn here. So citric acids get converted into oxaloacetic acid plus acetic acid. So here the enzyme citrate gets converted into pyruvic acid plus carbon dioxide. So this is the reaction happens. So these are the reagents and procedure. So this is the citrate positive bacteria, citrate negative bacteria. So the fifth one is catalase test. Here is the slide test. So inoculum drop plus 1 drop of hydrogen peroxide. So presence of bubbles shows the positive and no bubbles negative. So when we do on in the tube, so H2O2, so by adding bacterial culture we can see the bubbles or frothing seen. So yeah, this is the positive and this is the negative test. So these are the catalase positive and catalase negative bacteria. So coming to oxidase test, to identify aerobic bacteria with cytochrome enzyme oxidase with shown positive reaction for oxidase test. So here the positive deep purple color shows the oxidase positive test. So this is the principle and the reagents, and this is the procedure. Yeah, oxidase positive all the species of Pseudomonas and Vibrio species and Alkaligene species. So negative all numbers of Enterobacteriaceae, and so oxidase negative all members of Enterobacteriaceae and Stenotrophomonas maltophila. So coming to the methyl red test, so the positive test shows red color and the negative is yellow. So peptone gets converted into organic acid formation. So yeah, this is the principal reagent and procedure and the positive and negative bacteria. So the eighth one is Oaks-Prosker test or acetone production test. So yeah, this is a picture of Oaks-Prosker positive and this one negative. Pink color indicates Oaks-Prosker positive. So yeah, this is the principle, procedure and the interpretation. Yeah, so these are the positive and negative bacteria for Oaks-Prosker test. So estimation of pH of medium, 15th one is, so to estimate pH of a given media or solution using Lovie-Band bond comparator. So yeah, this is the principle and about Lovie-Band comparator. Yeah, this is the picture for that. This is the procedure. So coming to the Vidal test, so typhoid fever is an infectious disease caused by a bacteria called Salmonella typhi. So why are we doing this test? To detect the presence of Salmonella typhi. So yeah, to diagnose enteric fever such as typhoid. So the specimen we take is fresh fasting serum and the symptoms we see is low grade fever, diarrhea and headache. So coming to the test, Vidal test, here is this is the quantitative test. So O antigen, H antigen. So AH antigen, BH antigen. Yeah, for these 4 types we are doing this procedure. This is the control and these are the dilution titers. 1 is to 20, 1 is to 40, 1 is to 80, 1 is to 60 and 1 is to 320. Yeah, 1 ml of saline plus 1 drop of antigen. So this is the control. Coming to the titers, the first one for 1 is to 20 dilution we take a 1.9 ml saline plus 1 ml serum. So yeah, this is the dilution method. So yeah, 1 drop of respective antigen is to be added in each tube. So then we do incubation. So this is the qualitative test. So serum plus O antigen, serum plus H antigen, serum plus AH antigens plus BH antigen. So agglutination shows the positive, no agglutination shows negative. So yeah, this is the procedure. So ASO titer to diagnose rheumatic fever by ASO titer. This is the slide test. This is the dilution test. So it shows agglutination when positive. So this is the method procedure of a slide test. So the 18th one is VDRL, venereal disease of research laboratory to diagnose syphilis by VDRL test. So this is the one drop of serum plus 1 drop of antigen on VDRL slide. Mix and keep on rotator for 4 minutes. So this is a non-reactive, this is weakly reactive, this is reactive. So yeah, here is the principle and procedure. So this is the quantitative method of VDRL. So the 19th one is common skin test for tuberculin to diagnose tuberculosis with tuberculin test. So this is the principle procedure. So uses to measure prevalence of infection in a community to diagnose active infection in young children. It is used as an indicator of successful BCG vaccination. So coming to the bacterial examination of water to test the quality of water and quantity of organism in it. So observation of presumptive test. So acid and gas production. What are the concentrations we are taking? First one is double standard, and the second one is single strength, and third one is single strength 0.1 ml. Here we are taking 1 ml. So this is the principle and the procedure. So presumptive test or most probable number test. So this is the composition for eosin-methylene blue agar and indose agar. The second one is confirmed test. So let's see in picture form microbiological examination of water. So standard plate count. The second one is coliform test. Third one is membrane filter method. So bring the water sample in sterile container, serially diluted at 10 to the power of -4. So incubate at certain temperature. So CFU per ml. So this is the formula. And yeah, method can be done to know efficiency of purification of water. So yeah, the third one is completed test. So this is the water sample in presumptive test. First method, what are we doing? This is the lactose broth. When incubate the tubes for 24 to 48 hours at 37 degrees centigrade. So these are the Durham's tubes, inverted Durham's tubes. Here we are taking 10 ml, 1 ml and 0.1 ml. So we can see the acid production. Then gas and acid production. This is the positive and this is negative. No gas, no acid production. So the second one is confirmatory test, where we are using indose or eosin methylene blue agar. So where we can see metallic sheen colonies. Here lactose bile fermentation broth, brilliant green lactose bile fermentation broth. So we can see the gas production. So this is a confirmatory test. This is the second one. And coming to the third one, complete test. When we take these metallic sheen colonies, when we inoculate on nutrient agar, we can see the pink pointed colonies, whereas in lactose broth we can see the gas plus acid production. So microscopic observation gives that gram negative non-capsulated non-motile sporulating rods, example E. coli. So these picture form of microbiological examination of water. The first one is presumptive, second one is confirmatory, and third one is complete test. So the 21st one is bacteriological examination of milk. So testing of milk by methylene blue reduction test. So this is the principle and the procedure. So yeah, to detect number of microorganisms present in different milk samples by their ability to reduce the dye. Yeah, this is the picture. So time of decolorization. So 0 to 30 minutes, very poor quality. So this is the number of presence of organism. So within 30 to 120 minutes, poor quality. 120 to 130 minutes is fair quality, and 360 to 400 minutes good quality. So this is the sample A, this is sample B. We took raw milk and this is the pasteurized milk. So there was, in raw milk it was colorless and there was no color change. So duration is 6 to 8 hours is good. The quality of milk is good. So less than 2 hours is poor. So as the raw milk changes color blue to colorless within 30 minutes is poor quality. So pasteurized milk changes blue color to colorless within 6 to 8 hours, it is good quality. So yeah, so bacteriological examination of air to detect the presence of pathogen in air. So this is the principle, this is the procedure. So we use here is settle plate method. So coming to the examination of stool. So yeah, this is a collection of stool specimen, precautions taken after collection. So what are the physical examination we do? So microscopic examination of stool, what is the procedure? So saline specimen preparation and iodine specimen preparation. So the concentration techniques, if the presence of ova is small in number, they are not detected. So if the presence of ova is small in number, they are not detected by direct examination. So the concentration method is useful to detect these ova. This is the principle, this is the procedure. And what are the blood parasites? Are the organisms present in the blood. So these parasites are either protozoas. Parasites include Plasmodium leishmania, Microfilaria, Trypanosomas and Spirochetes. Yeah. So sample collection and preparation of blood smear. Yeah. So Plasmodium species, so it causes malaria in human being. So these are mainly of four types: Plasmodium vivax, malariae, falciparum and oval. So transmission happens with bite of female Anopheles mosquito. This is the life cycle and the diagnosis. Yeah, this is a picture of trophozoites and schizons. So erythrocytic stages of different species of Plasmodium gametocyte. So coming to Leishmania, three species: Leishmania donovani, tropica, and brasiliensis. So brasiliensis. So this the vectors are bites on sand flies. So incubation period is 2 to 4 months. Yeah, this is a picture of amastigote stage, and this is a promastigote stage. So coming to microfilaria. So microfilaria of Eukarya bancrofti. Eukarya malaria, loa loa, and monocella ozardi. So this is a picture of microfilaria of Eukarya bancrofti. So be prepared for all the biochemical reactions. So practice thoroughly for all the reactions and the principles. And yeah, what are the bacteria are positive and negative, so don't get confused with that. So make a clear chart of that and compare, and then you can remember well. So these are the mainly viva questions, and also yeah, so thoroughly prepare for vital test too. So quantitative method and also qualitative. So yeah, be prepared for this. Also VDRL about rapid plasma reagent. So yeah, be prepared for this. Microbiological examination of water, presumptive test, confirmatory and complete test. So also get prepared for this, for practical examination. So coming to concentration techniques, be prepared for all these parasites like Plasmodium and Microfilaria and Leishmania. So yeah, so I'm making these videos for BSc MLT students. So yeah, do well. All the best for your practical exam. I hope this record will help you for you to write your microbiology record. Thank you.